guys, in this video we're going to be looking at DNA, the code of life. Our introduction is going to look at the revision of the cell structure, the cell and emphasis of the ribosome, cytoplasm, things like that. And then we're going to look at the nucleic acids being DNA and RNA. So after that, we are going to then go and look at the nucleic acids. Here in the introduction, I will then merge DNA and also look at RNA together all at once, so that we just cover a lot of ground in one video. And then we're going to emphasize on everything that is mentioned here. I did try to find a picture of a cell, a diagram of a picture that is easy on the eyes. So we're going to be mostly focusing on the cytoplasm, the nucleus, and the ribosome. And then here we have the cytoplasm being this jelly-like material in the cell. The orange that you see here, that's our cytoplasm. So basically, it houses the organelles being the nucleus, the ribosome, and other things that you find in there that are not necessary for us. We don't really need to know them. Yes, those are our organelles. So our cytoplasm, aside from housing organelles, it is also the site of metabolic processes such as protein synthesis, which is going to be very relevant to us grade 12s. So metabolic just means it's a fancy word for bodily processes. Just a fancy word. You don't really need to stress about it. Bodily processes. You don't need to stress about it. And then we come to synthesis. Synthesis basically just means we are making something. We are making something. It's a fancy word to say making something, It's a science, so they have to be fancy. So we are making proteins, basically. So then we move on to ribosomes. Ribosomes are the site of protein synthesis, where the real synthesizing of proteins happens, where we really make the proteins. In simple, site of protein synthesis. And then moving on to our nucleus. One of our special organelles. So our nucleus here is very important because this is the location of our DNA. This is where we find our DNA. We find it in the nucleus. We find our genetic code there. It is also the site of DNA replication, and it is also the site of transcription. Site of DNA replication. And So the nucleus, this is where, like I said, we find our DNA there. We are gonna talk about it further when we're talking about the location of DNA. We are gonna talk more about it. But it is also the site of DNA replication. Replication just means we are replicating something. We are making an exact duplicate of DNA, of a DNA molecule. Exact And then we come here. Transcription is one of the phases of protein synthesis. Protein synthesis is divided into two phases, being transcription and translation. Transcription is the first process and translation is the second one. I'm gonna teach you a trick that is gonna help you remember which one comes first and which one comes second, if that is gonna give you a problem. Yes, so this is us on what we really need to know about the cell. Yes. We are now going to be looking at the two types of nucleic acids, being DNA and RNA. We have DNA, RNA. I'm gonna get you a diagram now to just help look at that. We are now looking at the DNA versus RNA. We have the ribonucleic acid, and then we have the deoxyribonucleic acid. This is where our abbreviation comes from. It comes from, our DNA abbreviation comes from the D, the N, and the A. Then here it's the R, the N, and the A. So the last two letters are the same, you just change the R and the D. Yes, ribonucleic acid and deoxyribonucleic acid. Further emphasizing on DNA and RNA, DNA forms the chromosomes in the nuclei of all living cells and carries the hereditary information of the organism. What this statement means is that you see this double helix structure, helix meaning coiled or twisted, comes together and forms this chromosome. They're tightly pegged together. These DNA helix structures, or structure, or whatever you want to call it, it comes together and just forms this structure called a chromosome. That's basically what it means. Hereditary information just means you inherit it from your parent, things like that. Hereditary means inheriting. Yeah, inherited information, sorry. So moving on to RNA. RNA is a single strand. If you notice, RNA is single and DNA is double. It's a single strand located in the nucleoplasm and cytoplasm. The RNA molecule is always a single strand of nucleotides. Here is the single strand. Nucleotide being a phosphate with a sugar and a nitrogenous base. That is a nucleotide. I will further explain that. Remember that the RNA contains of uracil instead of thymine. Again, those are our nitrogenous bases that I will further explain on. RNA is responsible for protein synthesis. We will then further explain it during the protein synthesis video. So to then make a difference table, we'll then make a table of differences here. Diving deep into DNA and RNA, these are their differences that will help you understand more. So their location, DNA is found in the nucleus, in the chloroplast, and in the mitochondria. We have DNA in the chloroplast, we have DNA in the mitochondria, and we have DNA in the nucleus. Whereas in RNA, we only find it in the nucleus and in the cytoplasm, found as tRNA, mRNA, rRNA. So here we have the 3 types of RNA, being messenger RNA, ribosomal RNA, and transfer RNA. They all look different. We are gonna emphasize more on messenger RNA during protein synthesis and transfer RNA during protein synthesis. Ribosomal RNA, we won't really look at it like that. We just mention it here when we talk about the types of RNA, but we don't emphasize on it. So this is how it looks. mRNA and tRNA looks like a letter T, like a bit of a letter T. So yeah. We'll talk more about the labeling when we are talking about protein synthesis so that it does not confuse us. See, transferring an amino acid, that's why it's called a transfer RNA. Messenger, because it carries a code. Yeah, this is a code. Those nitrogenous bases are a code. That is the message. The message is the code. Yeah. Moving on to the strands. DNA has a double strand, as you saw in the above picture, and then RNA has a single strand. Strand 1 and then strand 2. Here we only have one strand that goes like this, whereas here we have 2 strands, and then the second one is this one. So that is the double helix that they mean. And then here, sugars, we have deoxyribose in the DNA, the reason for the D, and then ribose in the RNA, for the reason for the R. This is what I was talking about before when I mentioned the DNA in the mitochondria, DNA in the nucleus, and DNA in the chloroplast. We have mitochondrial DNA, the type of DNA found only in the mitochondrion. And then we have nuclear DNA, the type of DNA found in the nucleus makes up genes on chromosomes. And then we have the chloroplast DNA, type of DNA found in the chloroplast. That's basically what it means. So yeah, when they say they can ask you and say, go in the animal cell, where do we find DNA? What is your answer? Your answer is in the mitochondria, and in the nucleus. Why? Because we do not have a chloroplast in the animal cell. As you can see, chloroplast, they then put it in brackets and say plants. You see? Right there, we have plants right there. Meaning it's only specific to plants. Yes. I was talking Greek before talking about nitrogenous bases. We have, these are nitrogen containing molecules: adenine, thymine, guanine, cytosine, and uracil. We have 5 nitrogenous bases in life sciences that we talk about. Each nucleic acid only contains 4 different nitrogenous bases. Uracil contains DNA contains adenine, thymine, guanine, and cytosine. That is DNA. And then we move on to RNA. RNA contains adenine, thymine, RNA contains adenine, guanine, cytosine, and uracil instead of thymine. You can't have uracil and thymine in the same nucleic acid. Point number 1. That's one thing you should know. And then we have what we call base pairing. Nitrogenous bases are paired together. That's how they move in a DNA molecule. Yes. Adenine always bonds with thymine, guanine with cytosine, to ensure the precision of DNA replication. We're gonna talk. These are structures that are made of 3 different parts. The parts being your phosphate, that is mostly represented as a circle, and then you have your sugar, that is mostly represented as a pentagon, and then you have your nitrogenous base, that is mostly represented as an arrow-like structure, something like that, a rectangular arrow-like structure, something like that. So as I mentioned before, that this is your phosphate. You just need to label it. You don't really need to know what exactly is going on, because one thing about textbooks, it will give you a lot of information that you will never need in an exam, trust me. This is your sugar, and then this is your nitrogenous base. This is just the general nucleotide. I will make I will draw a nucleotide that is specific to DNA and one that is specific to RNA. Phosphate remains the same in both DNA and RNA, doesn't change anything. But then here is something I want you to know here, that the nitrogenous bases that we can have in the possible nitrogenous bases that we can have in a DNA molecule are only 4. Can either be adenine, cytosine, thymine, and guanine. Whereas in RNA we have adenine, cytosine, uracil instead of thymine, and then we have guanine. And to finish off nucleic acids, we have the function of DNA and the function of RNA. Here the functions of DNA are: DNA makes up genes which carry hereditary information. Like I mentioned before, what hereditary information is. Genes. A gene is just a segment of DNA that codes for a particular protein. That's what a gene is. And then we have DNA. The second one is DNA contains coded information for protein synthesis. Like, it's basically saying that that pattern of nucleic acids and base pairing is a code for producing proteins, basically. That's what it means. And then the function of RNA is that it plays a role in protein synthesis. If you look carefully, both of them have to do with protein synthesis. So yes, this is directly from the exam guidelines. Sometimes your answers and your processes are exactly, they are needed. It's best to just look at them exactly from the guideline. And then we now move on to the history of DNA, which is mostly the discovery of DNA. We will need to know of at most 4 people here. You don't have to really know the dates, the years, because I've never really seen it in any question papers, specifically the years, but We have Rosalind Franklin. This is a woman, by the way. Yeah, we don't need to know that, but yeah. So Rosalind Franklin took X-ray photos of DNA with Morris Wilkins, her assistant. James Watson and Francis Crick formulated the double helix structure of DNA using a three-dimensional model. So another thing you will need to know under the discovery of DNA, when they ask you who discovered DNA, the answer is James Watson and Francis Crick. Watson and Crick. So yeah, most question papers don't even mention Rosalind. They'll just put it there just to confuse you, but that is never really the answer, which is a bit, yeah. So they then there were three of these scientists were awarded the Nobel Prize, being Morris Wilkins, James Watson, and Francis Crick. Rosalind Franklin ended up dying, so she was not nominated for that, and yeah, also, yeah, kind of because she was a woman, but yeah, she died of some type of cancer, I think. Yeah. Let me write it down. Watson, Crick, and Wilkins. So base pairing is about the pairing of nitrogenous bases that I mentioned before. So in DNA, this is how the pairing goes. DNA, adenine pairs with thymine. Guanine pairs with cytosine. And then in RNA, same thing, but we replace thymine with uracil. Adenine pairs with uracil. Guanine pairs with cytosine. So yeah, this is gonna be helpful during the couple of shows how nitrogenous bases pair. As you can see here, we have adenine, we have thymine, we have guanine, we have cytosine, and we have uracil. Here you see that the red pairs with the orange. You can see here that the red pairs with the orange, adenine and thymine. The blue pairs with the green right here, being guanine and cytosine. This is our DNA molecule. And then in our RNA molecule, there is no pairing that happens. Why? Because it's a single molecule. So in its natural state, there is no pairing. RNA is only helps during our translation, during protein synthesis. Yes, that is Greek for now. I just want you to remember it. And these nitrogenous bases are held by weak hydrogen bonds. To be specific, you have to say weak hydrogen bonds. When you look at this diagram properly, you can see that uracil, the purple, pairs with adenine, the red. So you can still see that this is still a single strand, right? Further emphasizing that this only occurs in what? In RNA. It's a single strand. Not that this is real. I don't think this is realistic. That as one strand connects with itself. I think it's just an example to show an example. Yes, held by weak hydrogen bonds. Yes. This is basically what goes on during base pairing. A plus T, adenine and thymine, guanine and cytosine, held by weak hydrogen bonds. And then in RNA, adenine and uracil, guanine and cytosine.